Hello everyone. Today I will introduce the paper submitted to IMATA. The title is Changes in Seminal Plasma Microecological Dynamics and the Mechanistic Impact of the Co-Metabolite Hexadecanamide in AZS patients. Firstly, I will introduce the background and the significance of this research. As we all know, from a worldwide perspective, infertility has surged to a prevalent rate of 15%. AZS is prevalent etiological factor contributing to male infertility. Emerging empirical evidence suggests that the male seminal microbiota is considered a key factor influencing the reproductive health of couples, pregnancy outcomes, and the well-being of offspring. Today, there exists a shortage of comprehensive and meticulously detailed analysis pertaining to the microecological dynamics of the seminal plasma in individuals reflecting with AZS. The dearth includes both the seminal microbiota and metabolite composition. The objective of this study is to delineate the composition of the seminal plasma microbial community and the metabolite profile, with a focus of a potential association with the occurrence of AZS. Firstly, we collected 120 seminal plasma samples based on the current international standard, with 30 samples in each group, including normal group, AZS-I group, AZS-II group, and AZS-III group. We used 16S rDNA sequencing and multi-omic analysis to conduct a comprehensive investigation of seminal microbial community and metabolites in AZS patients. As the result displayed, we found that there are significant differences in microbial composition within the seminal plasma between normal and AZS groups, especially 6 bacterial genera displayed in both figures. Subsequently, we conducted a difference analysis on their metabolite characteristics and found that there were also significant differences between the normal group and the AZS group, among which 3 different metabolites were the most eye-catching. After that, WGCNA analysis was used to further screen hexadecanamide, a differential metabolite closely related to AZS, and new samples were collected for targeted detection to verify the difference in the expression of this metabolite in different samples. Then we verified its effect with the help of in vitro and in vivo experiments, and found that it could enhance sperm motility. Subsequently, it was found that this metabolite could significantly increase the protein level of PAOX and CA2. Subsequently, we employed random forest analysis to predict the microbial and metabolic characteristics of the seminal sample within the 4 groups. As depicted in Figure A, we revealed that both AZS-II and AZS-III exhibit excellent diagnostic capability when compared to normal and AZS-I. Furthermore, we demonstrated that both AZS-II and AZS-III display favorable diagnostic performance in comparison to normal and AZS-I, with more robust diagnostic potential within the severe AZS group. Consequently, we identified 6 prominent genera in the seminal plasma of patients with moderate to severe AZS that display significant correlation with the metabolites, as depicted in Figure C. Finally, we summarize the findings of the article as follows. A. This study substantiates the potential clinical diagnostic utility of microbial composition and specific metabolites within seminal plasma for the AZS identification. B. Divergent microbial compositions and metabolite profiles within seminal plasma and discernible across varying degrees of AZS severity. C. Validation via in vitro and in vivo experiments corroborates hexadecanamide is as a crucial metabolite influencing sperm motility in AZS, coupled with the upregulation of target proteins. Accordingly, we formulate the conceptual hypothesis shown in this figure. In the onset of AZS, the presence of 6 genera of bacteria in the seminal plasma potentially induce dynamic changes that ultimately reduce the content of the hexadecanamide, which contributes to a subsequent decline in sperm motility.